from the genetics department at the Silverman Life Sciences Institute. Okay. Professor Benvenisty is spearheading the research of embryonic stem cells in Israel and is an authority worldwide. Thank you, please. Professor Benvenisty is heading today the stem cells unit in the Hebrew University, which is one of the largest embryonic stem cells research facilities in the region, and is head of a large research group, a very prolific one with a lot of interesting research and publications. And he's come to tell you today about the amazing world of embryonic stem cells. Nissim, please.
Thank you. Thank you. Thank you very much for the kind introduction. And I will try to tell you today, and first of all, welcome to Israel and to Jerusalem. I hope that you will be able to see the city beside hearing on, I hope, exciting research. So today I will tell you on, in my opinion, the frontier of medical research, which is human embryonic stem cells. As I will show you, there's a lot of interesting research, but at the same time, several ethical issues that are involved with these cell types. So I will start with what are embryonic stem cells and what are stem cells, and I will show you a short movie on the beginning of life. And then we'll touch, of course, on the issues of the ethics of stem cells, and specifically on the ethics of embryonic stem cells. And I will show you how different societies in Asia and outside Asia try to solve the issue of embryonic stem cells. Then we will discuss how we are going to use the cells, especially to differentiate them. And I will focus on the production of nerve cells, and even I will touch the issue of brain in a dish, and I will try to show you some very dramatic recent results using these cells. And later on, we will touch the issue of clinical trials. First of all, we'll touch the preclinical trials, and then the clinical trials that occur currently in the world with embryonic stem cells and the future of cell therapy. And we may be able to also talk about other issues. Let's see how much time we'll have. And please feel free to ask any question that you have. I would welcome a discussion. So what are embryonic stem cells? So basically, stem cells are the building blocks of our body. And stem cells have two different properties. So every cell that have those two properties is a stem cell. The first property is the ability to proliferate, and probably proliferate forever. And this is something that is unique to stem cells, but also you can see it also in cancer stem cells or in cancer cells. They have also the ability to differentiate, meaning to produce a more mature cell. So stem cell, a cell that have a capacity for self-renewal and a capacity to differentiate is a stem cell. And first of all, they differentiate to a progenitor cell. Progenitor cells still have a capacity to proliferate, but not for self-renewal like stem cells. And then, and only then, they become mature cells. Those are the cells in the skin, in the liver, in the lung. Those are the mature cells. So another way of looking at stem cells is their potency, whether they can differentiate only to one cell type. Those are unipotent, only one specific cell type. These are the cells in the germ cell of the male. The sperm, the stem cells in the sperm are becoming only sperm. Then we have multipotent stem cells. They can become several cell types. And pluripotent stem cells, pluri is like a lot or everything. Those are the cells that can become all the cells in our body. So we are really dividing, this is kind of introduction, the stem cells into two different types of cells. the adult stem cells, cells that are coming from our body, from the adult. They may come also from a fetus. But if it's coming from a tissue, the brain of the tissue, the fetus, the liver of the fetus, we call them adult stem cells because they are coming from tissues, even if they are fetal tissues. While embryonic stem cells are the pluripotent stem cells, the one that can differentiate to all cell types, and they are coming also only from a specific cell type during embryogenesis, and we'll touch that in a second. So the adult stem cells that we know, they are familiar very much, and we know a lot about it, that there was the first one that was isolated in humans, are bone marrow stem cells, the stem cells of the blood, and those are the stem cells that you use in bone marrow transplantation. And it is used for many, many different clinical problems. Among them are the bubble boys, the boys that do not have the immune system. So until you could transplant these stem cells into them, you have to have them in kind of a sterile bubble in order that they will survive, because otherwise they will die from infections. But now they can survive with a bone marrow transplantation. So all the adult stem cells can differentiate to specific cell type. The cells that can differentiate to all cell types are the pluripotent stem cells. And think about that one cell can become all the cells in our body. And luckily, as I will show you, we have these kind of cells in the lab, and we can actively grow them and study them and even transplant them into animals and into humans. So what are we going to do with human pluripotent stem cells? So there are many reasons to study human pluripotent stem cells. I will try to discuss only two types of research, cell therapy and disease modeling. And regarding cell therapy, so we are talking about disorders that they may benefit from cell therapy. When we usually talk about transplantation, we talk about organ transplantation, like kidney transplantation. But there are many, many disorders that you don't need to transplant a whole organ. Or you can't transplant a whole organ. Like in the brain, there is no sense of transplanting a brain. In Parkinson's disease, for example, you need to transplant the cells that are missing in those patients. And we'll discuss that later on. And it is true for other disorders that they will benefit only from transplantation of cells and not from transplantation of the whole organ. And this is true for diabetes mellitus, where you are transplanting the cells that are missing, the beta cells in the pancreas, cardiomyopathies, disorders of the cardiac muscle, the heart muscle, and then you don't have to transplant the whole heart, you just have to transplant the cells that are missing, the cardiac muscle cells. Or hepatic failure. Again, you don't need to transplant the whole liver. You may need only to transplant the liver cells, the hepatocytes. But where are you going to get those cells? So stem cells, the cells that renew themselves, are the source of these cells. And Science, which is the most important journal in the scientific community, claimed several years ago that approximately 3,000 Americans die every day from diseases that in the future may be treated with embryonic stem cells derived cells. So clearly there is a huge clinical need if we will be able to use these cells in the clinic, because there are many, many disorders that can benefit only from cell therapy using stem cells, either embryonic stem cells or other cell types. And again, science each year is choosing the breakthrough of the year, every year at the end of the year. And it is doing so in many, many topics of science: physics, astronomy, mathematics, chemistry, and of course biology. And in biology, there are many, many topics: neurobiology, botany, and so on. Twice in the past 12 years, stem cells got the breakthrough of the year. I'll show you the second time, but the first time was about 12 years ago, or maybe a little bit more than that, about 13 years ago, when human embryonic stem cells were discovered. So human embryonic stem cells are cells that are isolated from blastocyst stage embryo. Blastocyst is a structure of the embryo. In the human, it's about 5-day-old embryo. I will show you a movie sequence in the next slide so you will understand. But basically, and I will show you another movie showing how we are taking this human blastocyst and making human embryonic stem cells. But basically, all the research is done on IVF embryo, on in vitro fertilization embryo. So you have a sperm and an egg in a tube, and you have a zygote in a tube, and then it will make a blastocyst stage, about 100 cell stage embryo in culture, and this is the stage before it is transplanted or implanted into the uterus. And those embryonic blastocysts can either go into the uterus and produce a whole embryo, or if there are surplus embryos or disease embryos, this is something that we work in the lab, disease embryos, disease blastocysts that you don't want to transplant, we can derive embryonic stem cells from them with the permission of the couple. So this movie, the first movie that I'm going to show you, will show you the human development in less than a minute. So we are starting from one cell, and it's dividing quite rapidly, 2 cells, 4 cells, 8 cells, and then it's becoming a blastocyst. So when you starting to have this hole, and at the outer layer, you will have the cells that will become the placenta, and in the middle, you will have what we call the inner cell mass, a mass of cells that will become the whole embryo. At that stage, all the cells in this region are the same. All of them have the same capacity to become the whole embryo. At this stage, the embryo is still, it started, as you know, fertilization occurs normally in the fallopian tube, which is a tube. And then the embryo is moving from the fallopian tube to the uterus, which is again, it's in a cavity. It's not in a tissue itself. And only after it's becoming a blastocyst, it will be implanted into the uterus. This is the uterine wall, and you will see how the embryo is going in. All of those are extra embryonic tissues. Only this one will be the embryo. It will be clear in a second. This is the yolk sac. This is the placenta. Only this one, this region, will become the embryo. You may discover that it's already had this hand, an eye, and it's developing at the beginning very, very rapidly in humans and in other animals. I think that by now you can recognize that its identity. And amazingly, the first steps, and I will show you the movie again when I'm discussing it. So it's starting as one cell. After 5 days, we have about 100 cells. This is the blastocyst stage. We can do that. Until that stage, we can do it only also in vitro. After this stage, we cannot grow it in vitro. We can have to transplant it or use it for other purposes. And in vivo, the first stages, how the embryo is becoming the different tissues. First of all, we are having 3 types of cells, the outer layer, the inner layer, and the middle, and then it's becoming the whole, about 200 or 300 cells that comprise the whole body. This is happening extremely fast in human. After 6 weeks or 8 weeks, most of the differentiation already occurs. And then there is maturation of this embryo. But all the important things already occurred very, very early. So basically, we are starting with in vitro fertilization oocyte. We have an inner cell mass, and we are making embryonic stem cells. This is, again, few slides that are showing you this is the blastocyst, a human blastocyst. The inner cell mass is already out of the blastocyst. You will see that in a second in the blastocyst. There is one pipette that is holding this blastocyst, and the other one will try to take out the inner cell mass out of the human blastocyst. And as you see it, the inner cell mass, and we are taking out the inner cell mass, and then we can grow it, and we will get embryonic stem cells. And those are embryonic stem cells. This is a colony of about 150, 200 cells. Those are the tiniest cells that we know of. So when you look at that, this is the size of a normal cell. This is a fibroblast. This is another cell. You can see its nuclei, its nucleus, and the structure of the cell. And look at embryonic stem cell. This is one cell, another cell, another cell. They are the tiniest cells that we know of, and the most fast-growing cell. There is no other cell that are normal cells that are so small and grow so fast. And amazingly, only these cells have the capacity, as I will show you in a second, to become all the cells in our body. So clearly, there is a lot of promise, and I will discuss the research and the promise in a second. But before we are talking about the promise and the research, we have to discuss the ethics, because we are dealing with a human embryo. And different societies look differently on what we can do. So clearly, in all societies, the use of these human embryos is on embryos that otherwise will be discarded. This will be thrown away, will not be used for other purposes. So some societies will use surplus embryos, the one that were made, many were implanted in the uterus. If the couple finish their family plan, those embryos eventually that are surplus are thrown away. Instead of discarding them, they can volunteer to donate them for research. The other source, as I told you, we are doing genetic analysis. I'll show you that at the end of my presentation. We are doing genetic analysis at early embryogenesis, at early stages, at the 8-cell stage. And at that stage, we can tell, even at 8-cell stage, whether the embryo is sick or healthy. And the sick embryos are never transplanted back to the uterus. Those can be donated also for research. So those are the sources, and I will briefly discuss that. So different societies from all over the world are looking differently at human embryonic stem cells. In Israel, like in Japan and Singapore, and of course Canada and Spain and other countries, will allow the research with human embryonic stem cells, and we will allow the derivation of human embryonic stem cells, but only from spare embryos from genetic analysis or spare embryos from fertilization, from problem in fertilization. There are countries, including the UK, the United Kingdom, United States, with private funding, China, South Korea, and India, that will allow this research, but also to derive human embryonic stem cells from embryos that are done or are generated only from research, meaning that they can take an oocyte and a sperm and make an embryo just for research. Most of the world will not allow that, but there are some societies, like in the United Kingdom and in many places in the United States, as I will show you in a second, that will allow that. There are societies that will not allow any research with human embryonic stem cells, not the derivation and not the research. And this is true for Ireland and Poland. Germany, this is an outdated slide, and I'm showing you this outdated to show you how the world become more liberal. Germany will now allow the research of human embryonic stem cells, and it is now in this category, but not the derivation. France moved to this region, to this section, Switzerland as well, Austria as well, and Norway is on the way to move to this section, like Israel and most of the Western world, and Japan, Singapore, and others, and also Brazil. So most of the world is becoming more liberal. In Italy, this is the only case that since 2001, they changed the regulation and becoming more restrictive. And they will allow only the research, but not the derivation. But most of the world is becoming more liberal. So this is the map of the world, where you see this kind of orange. This is kind of permissive flexible regulation that will allow the derivation of the cells, but only from surplus embryos. The more dark brown are the countries, especially the UK, that will allow also the derivation of cells just for research. Japan is in this category, but it's kind of, again, an old-fashioned. They are in light, in kind of orange color. In the United States, it's the most absurd, in my opinion, condition, because different states in the United States will allow different regulation. So most of the states will allow the research with human embryonic stem cells. The red states will not allow any research with human embryonic stem cells, not just derivation. Any research is not allowed. And South Dakota and North Dakota are among these states. While in California, in Massachusetts, and in New York, the green states will allow all research, including some forms of cloning, and including making embryos just for research. So it's very dichotomic in the United States. In Israel, the regulation, and in that sense, the Jewish view and the Muslim view is very, very similar. They will allow the research. The Israel regulation is not based on the Jewish view. It's based on a notion that was developed about 20 years ago in the UK, that if a human being is dead when his brain is dead, so it's becoming alive when he starts to have a primitive brain, the neural tube. So the notion is that only at the second week of life, after 14 days, when you start to have a primitive, the beginning of a primitive brain, you are called a human being. This is the medical view. And this is what the Israel law is based. But the Jewish view is much more liberal than this view. The Jewish view is showing, is such that until day 40, the embryo has a status, but it's a lower status than a human being. And I will not go into details. It is based on research that was done in Egypt about 2,000 years ago, and it's based on the morphology of the embryo. If you look at that, the embryo, and this is the embryo at day 41, this is the day 28, and this is day 56. Until day 40, it looks like a worm. So at that time, they thought that it's becoming human only after day 40. And that is how, what it is based. And this is true for the Muslim and the Jewish view. So in a way, we got kind of an understanding in which societies we can do what research. But there was another revelation in the study of pluripotent stem cells that is now solving the ethical issues completely. And this is the other breakthrough of the year that happened about 4 years ago or so. And again, this was the breakthrough of the year with pluripotent stem cells, and in this case, it was done in Japan. You may know that Yamanaka did a very investigator in Japan that did an amazing research where he took somatic cells, cells from the skin, for example, and by simple genetic manipulation, they became pluripotent stem cells. So most of the ethical, not all the ethical issues, but most of the ethical issues with pluripotent stem cells is now solved because you don't need embryos for this research. You can use somatic cells and make from them pluripotent stem cells. So enough about the ethics, and now we can compare the embryonic stem cells to the induced. And let's go to the research. And the simple idea is that if you have stem cells from different tissues, you'll be able to transplant them. But it's amazing that for most of the cells in our body, on most of the organs in our body, although we had stem cells and we know that they either have stem cell right now or they had them during early development, we are not familiar. We are not familiar with the stem cells in the pancreas. We are not familiar with the stem cells in the kidney and many other tissues. So the simple idea is that if we have a cell that can make all cells, we may make all the cells in a dish and then transplant these cells to different patients. So in order to do that, we first of all made embryoid bodies. As you can see, there was a problem in this version of the computer, but never mind. So we are making embryoid bodies. Embryoid bodies are a fascinating structure. So you are starting with cells that are growing in a dish, and you can detach them, and then what you will see that they will make kind of balls of cells, and this ball of cells floating in culture will make kind of balloons filled of fluid, and they are floating in the medium. And in these floating embryoid bodies that you can see even without a microscope, you have all the cells, the different cells, the brain cells, the liver cells, the blood cells, but also the heart muscle cell. And this is an example of an embryoid body. This is the first embryoid body that showed such a function, and it was made in our lab many years ago. And what you can see is a human embryoid body where it's pulsating in rhythm. And it has many different cell types. Among them are cardiac muscle cells. And pulsating in rhythm because they are also pacemakers that telling this embryoid body to pulsate. If you look very carefully, you will see that it has a small arrhythmia. So actually there are two pacemakers that are pushing the cells to pulsate in rhythm. And that was kind of showing you that we are not just getting cells that look like brain cells or heart muscle cells, they are also functional. And this is now used for many purposes. One purpose is for screening of drugs. And as you know, there are drugs that are important to facilitate the heartbeat, and there are drugs that are important to slow our heartbeat. And those drugs are working, may not work on animals because there are differences between human and animal heart muscle cells. But those drugs can be now tested in those embryoid bodies, and when you add adrenaline, you will see it's pulsating very quickly, and if you have a beta blocker, it will pulsate very slowly. So basically, we and later others could show that you can make many, many different cell types, probably all the cell types in our body using embryonic stem cells. But more importantly, we don't want a mixture. We want one cell type. So if we want to cure a brain disease, we want only brain cells. And it is also true for heart muscle cells, blood cells. So we are trying to direct the differentiation and have enrichment or even purity of a specific cell type by growing the cells in different conditions. So if you have one factor, you may have more brain cells. And if you have another factor, you will have more heart cells. And this is the way that we are controlling this differentiation. So this is an example how we are starting with cells that are the tiniest cells in our body, and we can get neurons with long protrusions. And this is an example of neural cells made from embryonic stem cells that have these protrusions. And those can be used, as we will discuss, to treat different pathologies. But I would like to touch another issue. So we are talking about differentiation. But during development, we don't have just differentiation. Our brain is not just neural cells, just a mesh of neural cells. It's a tube. As you know, our brain started as a tube. Our spinal cord is still a tube with fluid inside it. The ventricles are evidence for such a tube. So the brain started as a neural plate, and then it's becoming a tube. And the whole brain is kind of a tube. So we can make neural cells, but can we make a tube of neural cells? Can we make pattern that as we see in the embryo? And to do this experiment, we had to push the research a little bit more, as you will see in a second. So one idea was that the signals for patterning are in the embryo. So if we put these cells in the embryo, whether these cells have the capacity to make a tube, not just a cluster of cells. And to do that, we made a chimera between human cells and animal cells. And chimera, and I will discuss the difference between chimera and a hybrid. So both of them, chimera and hybrid, are two things that we usually try to avoid in research, especially mixing genetic material from human and animals is something that we try to avoid. And I will show you that we needed these experiments in two types of our research, and I will show you the data in a second. So a hybrid is when you take an egg from one animal and sperm from another one. So all the cells in the body of this animal, half of the genetic material is one animal and the other one is another animal. And this is the hybrid. You are familiar with a mule, which is a hybrid between a horse and a donkey. There are other hybrids that you can see in nature. Hybrids can happen in nature. If you have a lion and a tigress, you can have a liger. And this is the largest cat that we know of. If you take the male is a tiger and the female is a lioness, it will be a tigon. But both of them are hybrids.
What happens if you combine the tigon and a liger?
Sorry?
What happens if you combine a tiger and a tigon?
Okay.
And a tigon?
So as you all know, in many, many parts of the world, you can see a mule, and actually the combination of a donkey and a horse is either a hinny or a mule. They look different, and it depends which is the mother and which is the father. And that occurred very commonly in many places in the world. A tiger and a lion occurred naturally, but in safaris. That way you can get this kind of hybrids.
So what happens if you combine a liger and a tiger?
Okay, both of them are sterile. All hybrids are sterile. So you cannot mate between those two. Okay. A chimera is something different. A chimera is when you take cells from one animal and combine it with cells from another animal. That do not occur between different animals. It's only occur in the lab. And many years ago, there was such a chimera between a sheep and a goat called Gip. Not a very attractive look, but still it survived. But there are also, it's not just curiosity. You can learn a lot from these chimeras, and especially from the chimeras between the quail and the chick. And in this case, they took part of the neural tube, part of the nervous system of the quail, and put it in the chick. And they got, as you can see, this is the chick, and this is part of the skin, or the pigment cells in the skin, but also the nervous system were from the quail. And some of these chick quail start to sing like a quail. So you can learn where is the center that is important for these functions. So you can learn a lot from chimeras, but these chimeras are regulated very clearly in order not to make, so there will be justification for these chimeras. So back to our question. Can we have, instead of just cells, a tube from human embryonic stem cells? And for that, we took human cells and we put them in the chick embryo. This is a simple experiment. You take an egg, you expose the embryo, and you transplant the human cells into the chick embryo. And in this case, we put these cells next to the neural tube. This experiment lasts just a few hours because the development of the chick is very, very quick. And it's about a one-day experiment. But what we got, so this is the neural tube of the chick. It doesn't look like a tube, but this is the middle of the tube. But this is the tube of the chick, the neural tube. What you see here are cells from human origin that became a tube. And became the tube because there were cues from the embryo, from the chick embryo, especially from this structure called the notochord. This is a structure that vanishes upon development. It appears only in the embryo and then disappears. But this structure is telling the neural tube to become a neural tube, and also probably to the human cells to becoming a tube. So we could show for the first time that we can get a tube and not just cells. But that was a proof of concept. Once we understood that we can do that, we didn't have to go back to make this kind of a mixture between the human cells and the chick, because we learned how to do that also in vitro. And by adding the right cues, we can now have this neural tube in a dish. And we don't need these cues from the embryo. We discovered these cues, and we can now use them. So what you see here is a tube, but it's a very symmetric tube. Look at the neural tube of the chick. So if you look left to right, it looks very symmetric. But what about anterior posterior? Here is the floor plate, and this is the roof plate. And it looks very different. So the neural tube has patterning of anterior posterior. Our neural tube was symmetric. So it became patterned, but it didn't become fully patterned. So it didn't get all the signals of up and down. So we asked ourselves, why don't we have these signals? What signals are we missing in order to have this pattern formation? And we asked ourselves, what do you need for patterning? And that was, part of that question was answered about 80 years ago and then Spemann got a Nobel Prize. Mangold, Hilda Mangold, was his PhD student. They did this magnificent experiment with it. Many of you probably learned about many years ago. Hilda Mangold eventually died at very early age. She didn't even see the final publication from an accident at her home, and she didn't get, of course, the Nobel Prize, but she was part of this research, and the research was amazing. The research suggested that in the embryo, there are some cells that telling the whole body to become either head or tail. And these cells, if you just move them, you will get two heads. That was the hypothesis. And they showed it very, very clearly by taking the cells that they suspected that those are what they call the organizer cells, put them in the frog, and now got two heads instead of one. Because they had two organizers, and now you can get two heads. So is it relevant to humans? So unfortunately, there are rare cases where you have diencephaly in human. This is not a siamic twins. Siamic twins are fused embryos, where you have two embryos and they are fused. These embryos are diencephalic, meaning that they have two neural tubes, and probably they have two organizers. This is very rare, and only few can survive to term, but there are cases that even can get into adult life. This suggests that even in human, you have an organizer. But this organizer was never discovered in human, and even in the mouse, we do not know much about it. So we asked ourselves, so what is an organizer? An organizer, to simplify that, is a structure that allows the secretion of some kind of a factor. So the tissues that are adjacent to this organizer will get a lot, and the tissues that are far away will get little. And that's how you can get, in a simple way, what will be the head and what will be the tail. It's a gradient. So we asked ourselves, do we have these organizer cells in our embryoid bodies? In our embryoid bodies, we have all tissues. Are we missing these organizer cells? So when we are getting this kind of differentiation, kind of chaotic differentiation, is it because we are missing this organizer cell, or is it because we have too many of them? And if we have too many of them, each of them will try to make an organizer, will push the cells to differentiate, but there will be a chaotic differentiation. So that was the two possibilities. Are these embryoid bodies lacking an organizer or having too many of them? Yeah. Can you hear me? Okay. Okay. So if there is a problem, tell me. But basically, what we found out, and now we genetically labeled the organizer cells. You can see them now as pink cells, and clearly we have them in the embryoid bodies, but we have too many of them. So we showed by many, many molecular and cellular characterizations that we have an organizer. I think it's off again. I think that the battery is low. But how do we show eventually functionally that it's an organizer? It's expressing what we expect from an organizer. It looks like an organizer, but does it function as an organizer? So it doesn't work. The battery is low. It's dead. So I'll try to speak up. If you can't hear me, let me know. So basically, in order to show that we have an organizer cell, we took this organizer cell and went back to the frog embryo. And we really went back to the sperm experiment. But instead of putting the organizer from the frog, we now put the organizer from the human cells. And we showed that once we have putting these putative organizer cells, we now have 2 neural tubes. And we then selected by cell sorting the organizer cells, and we can show that instead of having 1 neural tube, 1 brain, they have 2 of them. And if you look by section, this is the native, the normal neural tube, and this is the aberrant one. And that was in order to show that indeed we could show that we have an organizer cells in human embryonic stem cells. So I will try to talk about now preclinical studies and clinical studies, and then we'll start to wrap up. And I hope that we'll have some questions as well. So basically, if we think about neurological disorders like Parkinson's disease, we can take the human embryonic stem cells and we can differentiate them. We can differentiate them into neurons. If it's for Parkinson's disease, we'll differentiate them into specific neurons that are missing in these patients, which are dopaminergic cells, and then we can transplant them. Yes?
Can you take stem cells which are not embryonic and transplant them into neuron?
Okay. So if you take blood stem cells, they will not make neurons. If you take liver stem cells, they will not make neurons. The only stem cells that can make neurons are either embryonic stem cells, and we know of neural stem cells. There are some stem cells that we are familiar with, and those are the neural stem cells. So you can do this experiment, but in order to have enough neural stem cells to do this type of experiment in humans, and it has done in humans, I didn't have time to show you that, you had to use neural cells from 6 aborted fetuses at 5 months old that were spontaneously aborted or medically aborted, and to collect these neural cells from all of these embryos in order to inject them into one patient. So we do not have enough of those cells.
So you can't make polysomnic stem cells from skin cells?
Okay, so you can do it either with embryonic stem cells or with human-induced pluripotent stem cells. So you can do it either with cells that are coming from the embryo, or you can take skin cells from the patient, make them pluripotent stem cells, and then differentiate them into neurons. This is the case. So basically, that experiment was done in mice which have Parkinson, and it was shown that these cells incorporated into the brain of the Parkinsonian rodents and in a way cured their pathology. Similar experiment has been done with heart problems in the pig. And again, you take pluripotent stem cells, either human embryonic stem cells or human-induced pluripotent stem cells. You make from them cardiac muscle cells, and it was transplanted into the heart of a pig and cured its arrhythmia. What about humans? So currently, there are 2 experiments. Some of them is kind of on hold, but it's still there are some patients that already were transplanted with spinal cord injury. So in this case, you take patients that had an accident, and it should be a recent accident between a week up to 3 weeks after the accident, and you make from pluripotent stem cells, in this case it was human embryonic stem cells, you make oligodendrocytes. Oligodendrocytes are cells in the brain that are protecting the brain cells, the neurons. They didn't transplant the neurons themselves. They transplanted the cells that can protect the neurons. And the idea is, if there is a paralysis. And the paralysis is because there is a concussion to the spinal cord. There is not a complete cut in the spinal cord, but there is an injury to the neurons, so then you can add oligodendrocyte and they will protect the neurons from dying. And it was transplanted into 3 patients so far, and we are waiting for some results. The other disorder that we already have clinical trials in the world are macular degeneration. This is a type of blindness. There are 2 types of clinical trials going on now in the world. One of them is on hereditary blindness in children, and the other one is blindness in old age. And what they are trying to transplant are retinal cells. Again, these cells are coming from human embryonic stem cells, and they already transplanted them to few patients. And already there was a report in the most important medical journal, the New England Journal of Medicine, that at least 1 patient his sight got better. We have to wait for more patients to see if it's repeating itself, but at least there is some hope. So when we are talking about clinical trials, there is the hope, but still there are also problems. We have to take care that the cells will survive and to prevent the immune rejection. So I will just discuss that very briefly. So basically, one of the problems of organ transplantation in general is once you transplanted the organ, it may be rejected. The idea is that now with human induced pluripotent stem cells, we can take cells from the patient, make from them pluripotent stem cells, and then differentiate the cells, and then inject them back into the patient. And then the genetic material in the cells and the genetic material in the patient is identical. So in that sense, as I showed in this slide, there are many, many strategies to reduce or prevent the rejection, but the most promising one is to take cells from the patient itself and genetically reprogram it to pluripotent stem cells and then differentiate them back to the patient. Almost the last topic that I would like to discuss is a word of caution, because the cells, when we differentiate the cells to brain cells, to heart cells, they are kind of normal cells. But the undifferentiated, the primitive cells, the one that I'm so fascinated by them, the cells that can make everything and can grow forever, can also make tumors. So if eventually we want to transplant only the differentiated cells, the brain cells, the heart muscle cells, and we want to get rid of the undifferentiated cells after differentiation. But if by mistake we'll also transplant the undifferentiated cells, then we can make a tumor. And I will not go into details, but basically there are many ways to prevent that, and we showed 3 different strategies, either before the transplantation or after the transplantation, that we can avoid getting these tumors. So I will not have time to discuss all that I wanted, and I'll just go to the last slide. If there will not be any questions, maybe we can go back, but I just want to summarize that really pluripotent stem cells, whether they are human embryonic stem cells or human-induced pluripotent stem cells coming from somatic cells, really change the way I showed you, especially on regenerative medicine and developmental biology, but they are changing the way that we are studying these genetic disorders, and we now screen drugs and issues of toxicity. They are really changing the way that we do the studies. About 10 years ago, they got the breakthrough of the year because of human embryonic stem cells. A few years ago, it was the induced pluripotent stem cells that really changed the world again. What will happen in 10 years, I think it's up to you. So If there are any questions, I'll be happy to address them. Yes.
If the stem cell will clone, if the stem cell will clone like a new cell that is not of the host, like a frog cell will learn to grow feathers, well, potentially a stem cell can become that kind of cell?
Okay. I'm not sure. Okay. So you are asking whether a stem cell from one tissue can become a stem cell from another tissue? So,
even if it's not connected to the...
Okay. So the beauty of pluripotent stem cells is that they can become all the tissues in our body. All the other stem cells, the adult stem cells, can make specific cell types. But it was shown recently that you can do transdifferentiation. So what we are doing really is taking, we can take a somatic cell, make a pluripotent stem cell from it, and then differentiate that to another cell type, okay? And this is reprogramming into pluripotent stem cell, and then we are making somatic cells. We can now, it has been shown that we can do that. This is called transdifferentiation. And we can take one cell and make another cell type. The efficiency is not that good, but still it can be done. So again, biology is much more flexible than what we thought, but once you have a somatic cell, it can make only very few cells. You can make it more potent by transdifferentiation or by going through pluripotency. Sorry?
Only if it has that information beforehand.
So the information is in our genome,
Okay.
and it's extremely flexible. So we can get rid of all the information that is surrounding the genome and make a new cell from it.
Can we make it to that? Can we also add information from a genome that's unrelated to us?
I didn't get this.
By theory, can we take a genome of an animal that's unrelated to us, say a frog, and let's say a bird, and then a human could grow, hypothetically, a human would grow feathers. Even if it doesn't have that original.
This is a kind of science fiction. The question is, where can we cross between animals to get, this is kind of science fiction. There are synthetic organisms now that are made in the lab, but they are very simple bacteria. Whether we can do multicellular organisms that are synthetic, it's currently a science fiction. But please.
From what I see is that you do the induced pluripotent stem cells from somatic stem cells, and you've done that in vitro fertilization. Can we somehow use that process of inducing pluripotent stem cells from somatic stem cells, or from somatic cells, or what you just said is
Differentiation.
And you let the virus do that. So you let a virus do that genetic reprogramming. So you just put in the virus to the patient, and it will differentiate without the using of in vitro. Without doing.
Okay, so if I got the question right, maybe the question was whether you can do the transdifferentiation in vivo. Yes. Was that the question? Okay.
Using a virus.
Yeah. So this experiment was done in mice by Doug Melton at Harvard, which is a very good friend of mine. And what it did, it turned pancreatic cells that are not beta cells to become beta cells just by putting the right genes, making transdifferentiation in vivo. So we infected the pancreas of the mouse, and then these non-beta cells became beta cells. So that was done. The power of pluripotent stem cells is that you can make all cells. This is the beauty of that, and the efficiency is very good. with transdifferentiation, the advantage that you can go very quickly from one cell to the other, but the efficiency is very low.
What specific processes determine the original differentiation in embryos? So when it's gone from the blastocyst phase, what processes happen to do that, its normal differentiation into heart cells?
Okay. This is the $1 million question. Now, in your age, it may be the $1 billion question. The issue of how we are differentiating from one cell to all cell type is something that we have very, very little information. We think that it's part of that is where the cells are. So if the cells, for example, are in the surface, they may see other factors that our cells are in the middle. So it may partly based on the exposure to the environment. We are thinking that this is making a huge impact. But it's amazing that we are taking embryonic stem cell, and every time that we are making embryoid bodies, we always get ectoderm, mesoderm, and endoderm. And it's about the same percentage we are getting from those, and we are doing that in vitro. So there is something intrinsic, not just the environment. There's something intrinsic during differentiation that these cells know that they can differentiate to multiple cell types, and they will differentiate always to all of those cell types at the same time. And we do not, and we are studying that in the lab, like others trying to study that. We have some clues, but we are very much ignorant on how it's happening. Please.
Just now you said that we can use stem cells into stem cells for patients. So can I know how you can make stem cells into stem cells, as stem cells are very special in human, as the enzymes are very different from stem cells?
Okay. So the beauty about reprogramming, that the cells forget completely, almost completely, I'll touch that in a second, almost completely what it was. So you are taking a skin cell and you are making from that pluripotent stem cells that are very, very similar to embryonic stem cells. And then you can turn it to neural cells. And they forget almost completely what they have been. I'm saying almost completely because there are experiments that showing that there is some memory. In that sense, we did it and others did it. We did it with beta cells in the pancreas. It's very difficult to make, for several reasons, to make beta cells from embryonic stem cells. It's very difficult. So what we took beta cells, we make from them pluripotent, and we said if they have a memory, their chromatin, their methylation, I'm sure that Professor Chaim Sider, Howard Sider, told you about methylation in chromatin. If they have some memory that they were beta cells, maybe it will be easier to make them back to beta cells. And that's what we showed. We showed that in human. Others have shown that also in mice. So there is some memory. But basically, the amazing thing that, and this is really a revelation that Yamanaka from Japan taught us several years ago, that you can take a cell and by only 4 factors, it will completely, almost completely forget what it was and became a new cell type that can make all the cells in our body. Please.
When taking embryonic stem cells, are they effective only for members of the same family? Or what?
Okay. So basically stem cells can be transplanted to all ethnic groups. The problem is the rejection. So eventually, you want to transplant cells that are genetically either identical or similar. So you will go to the one that either the member of the family or take the cells from the patient and make pluripotent stem cells from it. But basically, and the experiment that I showed you with clinical trials in the eye and in the spinal cord are done with non-related stem cells.
So in the
case of the human and the chick getting together to form a chimera, wasn't the human cell rejected because the number of the chromosomes in the human is not equal to that of a chick? And secondly, what can you comment about the sterility of the chimera? Are they sterile also, like as compared to the trigon and the ligon?
Okay, so chimeras are not sterile. Only the hybrid is sterile. Chimeras, and we know these chimeras from different mice, they are not sterile because their gametes can be either from this species or that species. The gametes are not a combination of two species like in hybrids. Regarding the issue of rejection, this experiment in the chick takes only a day and everything is an embryo. At that stage, there is no rejection. The rejection in any case does not involve the chromosome, but the proteins that the cells are seeing on their surface, are showing on their surface. So basically, you are right. There is a rejection when we inject human cells into mice, unless we use mice that do not have the immune system, and then we can transplant the cells. In these chick experiments, the whole experiment did not allow any rejection, and in any case, rejection in the chick at that stage is too early for rejection.
So basically, we are removing all the factors that would eliminate the growth of the human, that would allow the growth of the human tissue.
Exactly.
So, all the growth factors which are inhibiting the human tissue are removed before we place the human cell into the chick.
So the idea was to use the factors from the chick that allowed the differentiation of the chick neural tube in order to direct the human cells to differentiate also the neural tube from human. That's what we showed. Eventually, it was being shown by others that you can do the same thing in vitro by using the same growth factors or similar growth factors that the chick is using. Do we have more time? I'll be happy to.
4 minutes. Over there.
What about stem cells from the umbilical cord?
Okay. So umbilical cord are stem cells that are coming from the neonate, the newborn. They are adult stem cells, okay? They are coming from a tissue. In this case, it's the blood. So almost all the umbilical cord stem cells are hematopoietic stem cells or blood stem cells. In the umbilical cord, we also have another stem cell called mesenchymal stem cells. These cells can produce the bone or adipose tissue. So there are two different stem cells in the umbilical cord. Both of them are adult stem cells and not embryonic. Embryonic are only the one that you can isolate from the 5-day-old embryo. Yes?
What about from menstrual blood? Stem cells that have been recently isolated from menstrual blood, how do they get stem cells?
So in different tissues, we have stem cells, but in menstrual blood, we don't have much of stem cells. If you want to get real stem cells, you usually go to the bone marrow, or you can have other stem cells in our blood, in the circulating blood, by injecting specific drugs that will take out the stem cells from the bone marrow into the peripheral blood.
So which component of the menstrual blood actually gives rise to the stem cells? The endometrial lining or the disintegrated oocyte? Which part?
So, okay. So the stem cells, or especially the embryonic stem cells, are coming from the oocytes in the ovary. Then the embryo is implanted into the uterus. But it's not that the uterine or the uterus itself is giving the stem cells. The embryo that is coming from the ovary and the sperm is having the stem cells.
So in case of menstrual blood, there has been no conception and the oocyte it has disintegrated. So in that case, what gives rise to stem cells?
Okay, now I understand. Once the oocyte is not fertilized, it completely dies, and you don't have stem cells. So once it is not fertilized, it completely dies. There are rare cases where it will be self-fertilization. It's called parthenogenesis. It's another story that we make stem cells from this fertilized oocytes, but it's another story.
So I want to thank Professor Benvenisty again.